PharmaPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Labisia pumila has this long history, especially in Southeast Asia, used traditionally by women after childbirth.
And what's really interesting now is that science is starting to catch up, confirming some of these traditional uses. We're seeing evidence for anti-cancer, antioxidant, antimicrobial, anti-inflammatory properties, even potential as a phytoestrogen therapy.
But here's the challenge for us as scientists. While we know it has these properties, pinning down the exact chemical compounds responsible, that's been, well, a bit tricky.
We need more solid data linking specific phytochemical groups to the effects we see.
Precisely.
Yeah.
And there hasn't been a whole lot of work comparing the different varieties of the plant either.
Which leads us right into the research we're looking at today. It focuses on 3 common Malaysian varieties. There's L. pumila var, we can call it LPA.
That's the one with its sort of broader oval leaves.
Then L. pumila var, pumila with green leaves, LPPG, and the same variety but with red leaves, LPPR.
And that leaf color difference in the pumila variety is mostly down to sunlight exposure, apparently.
So this study we've analyzed, it directly compares the antioxidant profiles and the phytochemicals in these 3.
Yeah, and that's really valuable information, especially if you're thinking about developing herbal products. Knowing which variety packs the most punch for a specific effect is, well, it's crucial.
Okay, so methodology. How did the researchers start breaking down these plants chemically?
Well, they began with a fairly standard reflux extraction, just getting the crude extract from the leaves.
The basic starting point.
Right. Then, to make the analysis a bit more manageable, they used reversed phase, solid phase extraction, SPE.
SPE. Okay, that's about separating compounds based on polarity, isn't it?
Exactly. Think of it like a filter. They use different concentrations of methanol, 0%, 50%, and 100% to wash the compounds off the SPE column. Things that dissolve easily in water come off first with the 0% methanol, really oily stuff comes off last with 100%, and the intermediate polarity compounds come off with 50% methanol.
Got it. So you basically sort the molecules into different bins based on how polar they are. What did they measure after that separation?
So after getting these fractions, they ran a bunch of assays. They measured the total amount of phenolics, TPC, total flavonoids, TFC, and total terpenoids, TTC.
Kind of getting a broad overview of the main chemical classes.
Yes. And then they assessed antioxidant activity using different tests, DPPH, FRAP, and the grease assay. Each of these looks at antioxidant capacity from a slightly different angle, different chemical mechanisms.
Okay, let's talk results. What about the initial extraction yields in those total compound classes? Big differences.
The overall yield, the amount of stuff they extracted, was pretty similar across the 3 varieties, somewhere between roughly 11% and 14% by weight.
Pretty consistent there.
But when they look at the types of compounds, that's where things got interesting. LPA, the Alata variety, had the highest total phenolic content.
Okay.
Whereas LPPR, the red leaf pumila, was significantly higher in total flavonoids and total terpenoids.
How significant?
We're talking up to maybe 50% higher in some cases compared to the others.
Wow, okay, that's quite a difference. So LPPR is packed with flavonoids and terpenoids, while LPA leads in phenolics. That definitely suggests different strengths. How did this play out in the antioxidant tests?
Well, all 3 showed some activity in the DPPH assay that's a common measure of radical scavenging, but LPA, the phenolic-rich one, was the strongest performer there.
Makes sense. Phenolics are often good radical scavengers.
Right. But interestingly, LPPR, the one high in flavonoids and terpenoids, was better in the grease assay, which measures scavenging of nitrite radicals.
So different assays, different winners. It really drives home that antioxidant activity isn't just one single thing, is it?
Absolutely not. Different phytochemicals work through different mechanisms. Some might donate a hydrogen atom, that's the HA2 mechanism. Others might transfer an electron, the SLET mechanism. These results suggest different varieties rely on different compounds and maybe different mechanisms.
And the study looked deeper into this by comparing the crude extracts and those SPE fractions, right? Using the different assays.
Yes, they compared activity at a specific level of 70% inhibition. And what they found was pretty telling. The 50% methanol fractions, the ones with intermediate polarity compounds, generally have the best antioxidant capacity in the DPPH and FREP assays.
Which involve both HAT and CET mechanisms, potentially.
Exactly. But the 0% methanol fraction, the most polar stuff, and the 100% methanol fraction, the least polar, they showed better activity in the grease assay, quenching those nitrite radicals, which is more of a CET mechanism.
So the polarity separation really helped tease apart which types of compounds contribute to which antioxidant actions. Very neat.
It really does. And because LPA showed such strong overall antioxidant results, particularly in DPPH and FRAP, the researchers decided to focus on characterizing its phytochemicals in more detail.
Made sense to zoom in on the most promising one based on those initial tests. How did they do that?
They used HPLC, first high-performance liquid chromatography. The chromatograms confirmed that the SPE fractionation worked well. You could clearly see that the 50% methanol fraction of LPA had the highest concentration of compounds absorbing light at 254 nanometers.
Which is a common wavelength for detecting things like phenolics and flavonoids.
Exactly. Lots of aromatic rings absorbed there.
Okay, so HPLC confirms the 50% fraction is loaded. Then comes the mass spec, right? The UPLC MSMS. What did that reveal? This seems central to our listeners' interest.
This is where it gets really exciting, especially for the phytochemistry field. Using UPLC MSMS on that 50% methanol fraction of LPA, they were able to putatively detect several glycosylated flavones.
Putatively meaning strongly suggested, but maybe not 100% confirmed structure without a pure standard.
Correct. But the data is very suggestive. And the big news here is that this study represents the first report of orientin and its derivatives being found in Labisia pumila Varieties. Alata.
First report. That's always significant in natural products chemistry. Orientin itself is pretty well studied, isn't it? Known for bioactivities.
It is, yes. And they didn't just find orientin, they also tentatively identified other related C-glycosylated flavones, things like isofitexin, leucinin-2, isoflavonoid, and different glycosylated forms of orientin itself, like mono and di-C-glycosylated versions.
And how does the MSMS help identify these specifically? What are the clues?
It's about the fragmentation patterns. When you break these molecules apart in the mass spectrometer, C-glycosylated flavones tend to lose their sugar units in characteristic ways. They saw neutral losses of specific masses 60, 90, and 120 Daltons, which correspond to losing parts of C-hexacyl or C-pentosyl sugar moieties. It's like a structural fingerprint.
So finding these specific polyphenols, these C-glycosylated flavones, concentrated in that highly antioxidant 50% methanol fraction of LPA, it provides a really strong chemical explanation for why it performs so well in those assays.
Precisely. These compounds are excellent hydrogen atom or electron donors, which is exactly what you need to neutralize free radicals.
And connecting this back to known science, C-glycosylated flavones like orientin and isofitexin have shown a whole range of potential benefits in other studies, right? Things like anti-hyperglycemic, antioxidant, anti-inflammatory effects.
Yes, the paper cites several reviews and studies highlighting these activities. So finding them prominently in L. pumila var, Alata adds another layer of scientific support for its traditional uses in the Malay community.
Okay, but they didn't only look at the 50% fraction. What about the other ends of the polarity spectrum, the 0% and 100% methanol fractions of LPA? They used proton NMR for those.
They did. The NMR spectra for the 0% and 100% fractions were less complex than the 50% fraction. Fewer signals, suggesting maybe fewer major compounds or less diversity in those very polar and very nonpolar ranges.
And did they manage to identify anything specific using NMR?
They used software ACD labs to help interpret the spectra and search databases. Based on that, they putatively identified a rather complex-sounding molecule in the 0% fraction. N1, 2-fan-3-dihydro-1,4-phosbenzodioxin, 6-vis, 1-hydroxy-3, morphlin-4, propan-2-decanoamide.
Okay, definitely a mouthful.
With an 84% match score. And in the very non-polar 100% fraction, they found something called 413-DP2, ethylpenta-3, 401-oxypropan-2, oxymethylhexyl 112 down, with a very high 99% match.
Interesting structures. How reliable are those software-based NMR identifications generally?
Well, the software uses sophisticated algorithms, comparing the detailed NMR data, the chemical shifts, the coupling patterns against large libraries. It uses things like HOC codes, incremental calculations, even neural networks. The paper mentions the software typically has small average deviations, so a 99% match is quite confident. 84% is still pretty good suggestive evidence.
Right. So bringing this all back to our main theme, the role of optimized MS analysis in phytochemistry for a scientific audience, what's the big picture takeaway from this study?
I think this study is a perfect example of how powerful optimized UPLC MSMS is for rapidly characterizing really complex mixtures like you find in medicinal plants. Labisia pumila isn't simple chemically.
Not at all. Thousands of compounds, potentially.
Exactly. And the strategy they use, first simplifying the mixture with SBE fractionation, then applying a high sensitivity, high resolution technique like tandem mass spectrometry, it allows for a much more focused investigation. You can start linking specific groups of compounds, like those flavones in the 50% fraction, to specific activities like the antioxidant properties.
It's moving beyond just saying the plant is an antioxidant to saying these specific molecules concentrated in this type of fraction are likely major contributors to this antioxidant mechanism.
Precisely. And it emphasizes the optimization part. Further tweaking of the MS settings, ionization methods, collision energies for fragmentation could potentially reveal even more compounds. You could get even finer detail on the structures and maybe start building structure-activity relationship models just from this kind of data.
So this study isn't just about Labisia pumila, is it? It showcases a workflow.
Absolutely. It demonstrates a really effective workflow for phytochemical analysis that you could apply to countless other medicinal plants, especially ones that haven't been studied in this much detail. It combines classic techniques like extraction and fractionation with the power of modern, optimized analytical chemistry, particularly mass spec.
A way to unlock the chemistry of traditional medicines.
Yes, to understand their therapeutic potential on a much deeper molecular level.
Okay, let's wrap this up. To summarize our deep dive into this Labisia pumila research, the key finding seems to be that the Alata variety, LPA, shows superior antioxidant activity, especially linked to its higher phenolic content.
And critically, the study provides the first report of orientin and other related C-glycosylated flavones in this LPA variety, identified mainly using that optimized UPLC MSMS approach on the medium polarity fraction.
Plus, they putatively identified other quite different compounds in the very polar and very non-polar fractions using NMR.
Right, and for the scientific community, these findings are really valuable. They help build that crucial bridge between traditional use, observed pharmacological effects, and the specific underlying chemistry. It gives us concrete molecules to investigate further.
So a final thought for you, the listener, especially if you're in the scientific realm. This work on Labisia pumila highlights how much chemical diversity might still be untapped in the plant kingdom. As analytical techniques like mass spectrometry become even more powerful and optimized, what other medicinal plants, perhaps previously overlooked or poorly understood, might reveal entirely new classes of bioactive compounds? Where could the next breakthrough in natural product-derived therapies come from, unlocked by these advancing analytical tools?
Thanks for tuning in to Bloom's Cast, brought to you by Blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
PharmaPal.